It's great to see you all. So just to start today, and in keeping with the spirit of reconciliation, we at TERN acknowledge the traditional owners and custodians throughout Australia, New Zealand and all nations, and we honour their profound connections to land, water, biodiversity and culture, and pay our respects to their elders past and present and emerging. And today I'm joining you from Ngunnawal country, which includes the land where Australia's capital city, Canberra, is located. So again, thank you for joining us today. And for those that haven't met me, my name is Beryl Morris, and I'm the Director of TERN, Australia's Ecosystem Research Infrastructure. And in TERN, we do a lot of work to monitor critical parameters related to the environment and climate. And we wouldn't be able to do that without all of the funding and support that we get. And on this slide, you can see some of the people that we rely on, and we're very pleased to have their support. And of course, as we start our second webinar for 2023, we also want to thank you for joining us. And so we have something like 15 countries joining us today with about 300 registrants. So I hope you're all going to be subscribed to our newsletter so that we can stay in touch with you. So just a few housekeeping points before we get going. So for questions to the speakers, we hope that you use the Q&A button on the bottom of your screen or wherever it's located. And we'll answer them towards the end of the webinar. And if you have any technical issues, put those in the chat so that we can deal with those fairly quickly. And today we're recording the webinar so that you can revisit the talks, or you can share the link later with people that you would like to know about it. So in registering for this webinar, you would have seen the bios for our three speakers. And in the interest of time, I'm just going to give you a very brief introduction. So Professor Alan Anderson, spelt incorrectly there with an E end at the end, is from Charles Darwin University, where he's the head of research in excellence and impact, and he's also Professor of terrestrial invertebrates. Dr. Matthew Lott comes to us from the Australian Centre for Wildlife Genomics, which is at the Australian Museums Research Institute, and he is a scientific officer at that place and a geneticist. And our last speaker is Simon Robson, who's from Central University in the School of Health, Medical and Applied Sciences. And Simon's currently involved with TERN in a BAT project. So I'm now going to hand over to our first presenter, Alan, who's going to commence our presentations. And I'll be seeing you again later during the Q&A. So thank you very much.
Hello, everyone. Thank you for joining, and I'm really delighted to be here to speak with you about terrestrial invertebrates as bioindicators of ecosystem change. It's been a favourite topic of mine for nearly 40 years. So terrestrial invertebrates, well, why terrestrial invertebrates? Well, there's two good reasons. One is in terms of biodiversity. So yes, invertebrates, of course, don't have a backbone, but they're actually the backbone of biodiversity. And so in this picture here, the different creatures are scaled according to not their physical size, but how many species they represent. And it gives you a very, very different view of an ecosystem, what we're used to when, we're very impressed by big things. And so beetle here represents insects. And you can see it's by far the largest creature on this landscape. And here, down here under the mushroom is elephant, representing mammals. And so terrestrial invertebrates include all these other things that crawl along the ground, and together they make up the 95% of all the animal species. So, that's one really important thing to keep in mind. If we're interested in biodiversity, well, invertebrates are at first cut for all faunal biodiversity. And the second really important point is that these creatures play so critically important roles in our ecosystems. Every major ecosystem process is to a large degree influenced by invertebrates, and they're so well connected within an ecosystem, it means that if we know something about how the invertebrates are doing, that's telling us powerful information on how the broader ecosystem is going. And so I want to focus my talk on my favourite insect group, ants, and share some of the experiences I've had with using ants as bioindicators of ecosystem change. And ants are particularly good indicators for these four reasons. One is just their sheer ecological dominance. So just sheer faunal, their contribution to faunal global biomass is enormous. That's one important point. Another is that we know a lot about how ant communities work. And so we can predict how they respond to disturbance. And I'll give you some illustration of that later. And third, we've got some good data to show it's not just what ants are doing, but what ants are doing reflect what's happening in the broader ecosystem, which is, obviously of fundamental importance to be a bioindicator. And the final one is I'd like to share with you where we're at in terms of increasing the monitoring efficiency of ants and other terrestrial invertebrates, because it's no point having an invertebrate group or any bioindicator that might actually work, but if you can't sample it, if it's too difficult, then it's just not going to be useful. And so ants have a long history of being used as bioindicators in land management in Australia and overseas. Wide range of ecological impacts of different land uses, as I've got here, and in terms of ecosystem restoration as well. I want to focus, though, on some experiences I've had with using ants as bioindicators of the success of mine site rehabilitation. And I'm going to start off with a case study I've been involved with for decades at Ranger Uranium Mine, near where I'm sitting here in Darwin, on the far northern coast of Australia. And Ranger is a huge uranium mine. It's one of the biggest in the world, and it's highly contentious because it sits right within the boundaries of one of Australia's most iconic national parks, Kakadu National Park. And the requirement at Ranger is to restore the ecosystem after mining to an extent that the habitats are similar in all ways to those in surrounding Kakadu National Park, so a really high bar. And this series of pictures I've got here, this is the mine with just a pile of rocks being levelled out and then planted. And here's some revegetation after about 10 years. And up here, I've got a picture of what the surrounding landscape looks like as a reference condition. That's the goal, is to produce landscapes like this. And so the question is, well, how's that rehabilitation tracking? We can see that it's got some vegetation there, but in terms of broader ecosystem change, what's happening here? And so we've sampled ants at a number of sites, and I'm just showing you a picture here where the sites are positioned according to how similar they are. The more similar the sites are, the closer they are together. And you can see straight away that these black dots here, these are the reference sites. These are undisturbed sites adjacent to the mine, and that's where we're aiming to be. And you see they're clustered very close together because they're very similar to all the sites undergoing rehabilitation. And here's this sequence of a rehabilitation trajectory of a site that was a third of the year old, fourth of the year old, and so on, 2 years, 13 years. And you can see it's tracking sort of towards the reference sites, which is what we'd like to see, but it hasn't got there. And we've been able to provide feedback that the reason it hasn't got there is that they haven't quite got fire management right. And so they need to introduce fire management into their rehabilitation. The important point is that this is not just what ants are doing. As I mentioned before, it's providing a broader indication of how the ecosystem is doing. And so we've been able to calculate the extent to which ants are reflecting, first of all, the total invertebrate assemblages here, the soil, ground, vegetation, and then species level changes. And so these coefficients range from 0, which means there's absolutely no relationship, to 1, which is a perfect relationship. And so these are all positive, sometimes highly so, which means that ants are not giving a perfect description of what all these things are doing, but they're giving you a really good general indication of what terrestrial invertebrates more generally doing. They're also reflecting changes in some really important ecological processes. And here's one I'm showing some information on. And this is a relationship between the number of ant species here, this is from Ranger as well, and soil microbial biomass, which is really critically important for key functions in soil, including the capacity for nutrient cycling. And you can see that there's a strongly positive correlation between what ants are doing, this key ecosystem function, particularly nutrient cycling. I'm going to give you an example where we've had some extraordinary success in restoring a site after mining. And there's an example from Allied Mine in Queensland, so a couple of thousand kilometres from where I am. This is a dissimilarity tree where the lengths of these branches reflect the extent of dissimilarity. So the shorter the branches, more similar sites are. You can see the reference sites come out, I've got them in green, and here's what a reference site looks like, come out as being very different, all these rehabilitation sites. Yeah, here's an example of what one of these sites look like. Really interestingly, this rehabilitation site, TH91, falls out as clustering with the reference sites. And if you look at some of the detail of what ants are doing, if you look at either the mean number of species per sample, you can see if you compare the rehab sites, 22, the reference sites have twice as many. Well, this particular rehab site has got even more mean from the reference site, but way more than the other rehab sites. And if you look at particular indicator species, these species here are indicating reference conditions, so not occurring at all in the rehab sites, except for this particular rehab site, which is similar abundance to the reference sites. And conversely, ant species here that are indicative of the rehab sites, so most common in rehab sites and either absent or not at all common in the reference sites. It's the same with the site TH91. So whichever way you look at it, the site has restored to an extremely high level and is in fact is indistinguishable from the reference site. So that's a great success story. I want to talk more about what I was saying before, how we know the dynamics of ant communities so well that it can form a basis for us interpreting the changes that we see when we use ants as bioindicators of ecosystem change. So here's just a generalized model of how ant community dynamics work. And this is how environmental stress, disturbance interact with these different functional groups. So don't worry too much about all that detail, but here's an important piece of information that I want to share, in that we can classify ant communities in a similar way that everyone's familiar with classifying plant communities. And that's by recognizing these three major functional types in biological communities: dominant groups, subdominant, and ruderal. These ruderal things are those that respond positively to disturbance. And we all know that in plants, these are familiar plant functional types: trees, shrubs, grasses, the three major life forms, which form the basis of vegetation classification. So whether we're talking about forests or shrublands or grasslands or any combination of those, we use these major plant functional types as the basis for it. And the good news is that we can identify ants that play equivalent roles. And so the equivalent to dominant trees are these dominant ants within the subfamily Dolichodorini, which, like dominant trees, they exert a huge influence over a wide range from where they nest, and they're highly aggressive, and they have a big influence on what the rest of the ant communities do. And similarly, generalized myrmecines play a functionally similar role to shrubs, in that they are subdominant to the dominant Dolichodorini. And then similarly, we can identify opportunists within the ant communities that play a similar role to how grasses function within vegetation ecology. And then we can map all this on this triangle that shows you how things vary in relation to the extent to which community is influenced by disturbance, and disturbance defined as any factor that removes biomass, which is distinct from stress, which is any factor that limits the production of biomass, so limits productivity. When stress and disturbance are low, then competition is high. And so there's this relationship, and one can plot ant communities according to some diagram of the relative importance of disturbance, stress, and competition. These letters D, D, and GM refer to those main ant functional groups I mentioned, and the numbers 2 and 3 and 4 and so on reflect the relative abundances of them. And so, to give you an example, we can plot how in a tropical savanna, where I'm sitting in a tropical savanna landscape experiencing frequent fire, this is what the ant communities are typically classified as. As we exclude fire, it moves through these other communities, ends up in a very different space, a very different community. So we have this great background understanding of how ant communities work, which is a really powerful tool for interpreting what we find when we use ants as bioindicators. I want to finish by talking about how we need to improve the efficiency of which we can use invertebrate bioindicators, because I totally understand for most land managers, invertebrates are just beyond their experience, and there's so many species difficult to identify. And the more we can increase the efficiency which they can be used as bioindicators, then the more they're going to be applied to land management. And I'm going to talk about an example here I was involved with in Mount Isa. So here it is here, far northwest Queensland. At the time, this smelter here was the world's largest point source, sulfur dioxide. Here on this map is the town Mount Isa, and the winds come from the southeast this way, which means the impacts are all northwest of the mine, close to the mine, highest impacts, and reducing as you go further away from the mine. This is 30 kilometres from the mine. And then we can use sites downwind as a control, just to work out what's going on in terms of the biodiversity impacts beyond the smelter. And so I had a PhD student who did some very intensive ant surveys as part of this project. And this is what he came up with. So he spent a year doing this, so lots of ant species. And these are these 5 major findings: that the different habitats have distinct ant faunas, and that ant abundance and richness declines with the concentrations of sulfur dioxide. Species composition varies, and individual species show very distinctive patterns in relation to concentration of sulfur dioxide, which is distance from the smelter itself. And so what we did is we developed a really, really simplified protocol where instead of putting out specialist pitfall traps to catch ants, we just used the bycatch that was collected by our vertebrate survey team, who had these big bucket pitfall traps. So we just pulled the ants out from them. And then we thought, we'll only look at big ants. Make it a lot easier for people to collect and process, and we won't worry about how common they are, just whether they occurred there or not. And we found that by doing this, we reduced the lab effort by, well, I said more than 90%. It's more like 95%, 99%. I could do all this in 1 week, where it took my PhD student a whole year to do his intensive ant survey. And much to his dismay, I was able to recapitulate every one of his major findings, every one of them. And here's an example of how ant community composition varies systematically with the concentration of sulfur dioxide. So low concentrations close to the smelter, medium concentrations more distant, and then high concentration, the other way around, isn't it? High close through to low 30 kilometres away. And again, you can see that there's clear patterning in the ant communities according to that. We were able to detect an impact on ants using this highly simplified protocol, and impact more than 30 kilometres from the smelter. I want to just sort of finish it. This is the future of using ants and other invertebrates as bioindicators, and that is metabarcoding and eDNA. So instead of the traditional practices where you pull out all the specimens from your traps and you sort them into species and you curate them and you mount them up and you identify them, and very time-consuming process, you can just smash them all up, then extract the DNA, amplify the DNA, sequence them, then use and establish a library that will tell you what specimens are there. And then even if we're able just to take soil samples, even worry about collecting specimens, and then go through this procedure again. So we're currently doing a lot of work on just seeing how effective these genomics approaches are. So hopefully I've described how at least for ants, they're really good bioindicators. They tick all these boxes really well. But my main take-home message is this: vertebrates are powerful indicators of ecosystem change and a really valuable land management tool under all sorts of land use scenarios. So I'd like to finish there. So thanks very much for listening, and I'm going to hand over to Matt now.
Cool. Thanks very much for that, Alan. You're definitely a tough act to follow. So, just going to Alrighty. So, hi all, my name's Matthew Lott. I am a geneticist here at the Australian Centre for Wildlife Genomics down in Sydney, and I'm joining you today from Gadigal country. And I guess our name probably gives most of it away, but the ACWG basically uses genomics, by which we mean the study of all or part of an organism's DNA, to try and inform evidence-based management of Australian wildlife by exploring how things like the climate and the environment have shaped the unique evolutionary or demographic histories of species that are of significance either from a conservation or commercial perspective. And most of the data that I'm going to be presenting to you today came out of a study that was published last year in the Journal of Molecular Ecology, where we investigated the mechanisms underlying population structure and adaptive genetic variation in the koala, which is quite an iconic Australian marsupial that I'm guessing a lot of you will be familiar with. But today I'm going to be presenting those data in a slightly different context and asking the question, do they have implications that extend beyond the management of a single species? And does koala genomics or genomics more generally have any utility as a bioindicator to inform ecosystem level management? So let's first start really basic. What is DNA? Well, in a nutshell, DNA is just a large molecule, which is composed of two long chains of smaller molecules, which are known as nucleotides, that coil around each other to form that distinctive double helix shape that you're probably all familiar with. And as I'm sure everyone's aware, DNA carries the genetic instructions for the development, functioning, growth, and reproduction of all known organisms, with the possible exception of some viruses, depending on where you come down on them being living organisms or not. But what a lot of people might not realize is that DNA is actually a lot more than the blueprint for making a plant or an animal. It's also a record of how that organism has changed over time. And by comparing the DNA of different species or even different populations within the same species, we can start to gain an understanding of not just how those groups might have responded to ecological or climatic disturbances in the past, but we can even start to make predictions about how they will respond to current and emerging environmental challenges, including those that are being caused by humans. So by studying the DNA of koalas, for example, which is a widely distributed species across much of eastern and southern Australia, but increasingly less so in recent years, unfortunately, we can potentially do things like identify genetically distinct groups or populations of koalas across the continent. We can identify the physical features or characteristics of the environment that might have led to different groups of koalas becoming genetically distinct and the approximate time periods at which they might have diverged. We can assess demographic trends across time, so things like the changing number of breeding individuals in a population or the level of migration between neighbouring populations. And we might even be able to identify some of the environmental or anthropogenic selection pressures that are driving the emergence of new genetic variation in isolated populations. And by understanding these processes in koalas, we might in turn be able to gain some broader insights into the status of the Australian climate and environment across time. And that's clearly something that's incredibly important because honestly, it's kind of difficult to overstate how much trouble most native Australian ecosystems are currently in. So over the past 200 years, we've experienced a kind of perfect storm of rapid habitat destruction and introduced predators, herbivores, and pathogens that have resulted in a greater loss of biodiversity than in any other developed nation on the planet. And coupled with the emerging effects of anthropogenic climate change, these processes are causing substantial and ongoing shifts in the distribution and composition of Australian flora and fauna communities, which means that specialist folivores like koala are predicted to be among the fauna that are most heavily impacted by human-mediated environmental challenges in the near future. So while I certainly wouldn't necessarily make the argument that koalas are an ideal bioindicator, they do certainly share some characteristics of one in that they're an abundant and widely distributed species. Because they are specialist folivores, we can expect koala populations to display responses that are broadly proportional to the severity of environmental climatic disturbance. And they're also a charismatic species that enjoys a pretty significant amount of support among the public, which is, I think, critical for kind of maintaining those long-term biological monitoring programs. And that's why it might be useful to examine the DNA of koalas, not just from a population genetics perspective, but also from a bioindicator. Now, before I present some of our data, there's one other part of this equation that I wanted to quickly talk about, which is the importance of natural history collections for environmental monitoring and management. So rapid advancements in what we call high throughput next generation sequencing technologies are allowing researchers to retrieve genomic information from museum specimens. So things like old skins or residual tissue on skeletal elements that in some cases can be hundreds of years old. And they can be used to address questions that may not be possible using contemporary specimens alone. So for example, using museum specimens, we can not only establish the historical boundaries between genetically distinct populations or lineages in areas where a species may no longer exist, but we can also directly assess recent genetic responses to things like climate change and habitat loss over time. But historical specimens are not the only way that museums can contribute to threatened species conservation and ecosystem monitoring. So the Australian Museum specifically, for example, is the custodian of one of the largest, if not the largest, collection of koala frozen tissues in the world. So we regularly receive donations from veterinarians, wildlife carers, and researchers from across the country, which are then stored here in perpetuity using our specialised infrastructure for things like future research or monitoring projects. You can sort of see by how working at the Australian Museum, we were uniquely positioned to access a range of different sample types that allow us to address some really interesting questions about koala biology and ecology that are going to be foundational for developing more effective conservation strategies. So I'm not going to dwell too long on our methods, but basically we sequenced the protein coding gene regions or exons of around 260 koala specimens that represented a mix of both contemporary and historical samples. And these samples were sourced from around 92 locations that span the modern distribution of koala across eastern and southern Australia, and included specimens from areas where koalas are now recognised to be locally extinct, such as in northern Sydney. So from the exome sequence data, we were able to identify a number of biological markers called single nucleotide polymorphisms, or SNPs, that were then used for the various downstream analyses. And in addition to the exome work and in collaboration with researchers at the University of Copenhagen in Denmark, we also resequenced the entire genomes of 37 historic and 25 modern koala specimens. And these data were used to address some very specific questions about the historic population demographics of koalas, which I will get to in a moment. Okay, so on a continental scale, both model-based and multivariate clustering procedures supported the existence of 5 major genetic clusters or lineages of koalas. And what's more, most of these major genetic clusters appear to be separated by well-known Pleistocene biogeographic barriers. A biogeographic barrier just being a feature of the landscape that acts as an impediment of animals. And when you examine them together, you find several commonalities across these biogeographical barriers. For the most part, they are lower elevation zones, dry, warm, open woodland or grassland that provide suboptimal habitat for forest-adapted taxa, and that you would therefore expect to be pretty difficult for koalas to disperse across. So some of the more significant biogeographic barriers in koalas appear to be the Sydney Basin, the Clarence River Corridor, and either the Brisbane Valley or the St Lawrence Gap. It's a little hard to tell at present because there's a bit of a sampling gap in central Queensland where koalas are now very, very hard to find. And then one other thing that I just wanted to quickly draw your attention to before we move on is that part of what makes koala conservation so tricky is that these animals are subject to wildly different legislation depending on what part of the country they're in. And koalas have been declared endangered in Queensland, New South Wales, and the Australian Capital Territory, but not in Victoria and South Australia. So the reason I bring that up is that you'll notice that neither existing jurisdictional boundaries or management units actually reflect how genetic diversity is distributed between koalas. It doesn't map onto their actual population structure. And that can present quite a unique management challenge. So if this is how genetic diversity in koalas is currently distributed, I guess the next logical question would be what sequence of events might have led to that situation? And that's the question that's probably of more direct interest from the perspective of koala genomics as a potential bioindicator. So to investigate that, I used something called approximate Bayesian computation, or ABC. And in a nutshell, that little stick figure there is a graphical representation of a mathematical model which best describes the sequence of events which led to the distribution of genetic diversity that we currently observe in koalas. And it seems like that what probably happened was around 300,000 years ago, assuming an average koala generation time of about 7 years, the Southern Australia, Mid-Coast New South Wales and Queensland genetic clusters all emerged more or less simultaneously from a hypothetical ancestral population. And while you do need to take precise divergence times with a little bit of a grain of salt, these data strongly suggest that the demographic history of modern koalas has been heavily influenced by a major climatic reorganization that occurred around 430 to 300,000 years ago. That led to increased severity of the glacial interglacial cycles and drove progressive aridity across much of the Australian continent. So the more pronounced climatic fluctuations of the middle to late Pleistocene are in turn hypothesized to have caused cyclical range expansions and contractions in a lot of taxa, which you would now expect to present as divergent geographically discrete lineages, as predicted by refugia hypothesis. So a refugia just being an area that a threatened species is able to retreat to, persist in, and eventually re-expand from during periods of environmental disturbance like that caused by rapid climate change. So what our data are suggesting here is that there are at least 3 major refugia exploited by koalas during the Middle Pleistocene glacial cycles, and that the foundation of both the Southeast Queensland and South Coast New South Wales genetic clusters were driven by secondary contact between these previously isolated lineages, likely during periods of mesic forest expansion that was precipitated by the warmer and wetter interglacial periods. So these findings are also fairly well supported by independent bioclimatic modelling, which shows that at various points in the species' recent evolutionary history, for example, during the last glacial maximum, suitable koala habitat probably retracted to geographically restricted areas of Southeast Queensland, Northeast New South Wales, and small coastal areas of Western Australia, South Australia, and Victoria. And what's interesting is that the last glacial maximum only occurred around 26,000 years ago, whereas these genetic clusters all appear to be much older than that, which would indicate that these refugia were exploited by koalas across multiple Pleistocene glaciations. And the reason that that's now potentially concerning is that climate modelling indicates that suitable koala habitat is expected to go an eastward range contraction in response to anthropogenic climate change, which is not dissimilar to what occurred during the last glacial maximum. Only this time, their traditional refugia might not be available to them anymore because that is also where most of the human habitation of Australia is concentrated. And on this next slide, what I've got is an even more detailed breakdown of the demographic histories of different koala populations across time. So these data were based on the sequencing of the whole genomes that I mentioned, and they were analyzed using something called a pairwise sequentially Markovian coalescent method, or PSMC, which basically uses the genome sequence of a single individual or individuals to estimate demographic history covering a time span of thousands of generations. And the way you read this is that on the x-axis, you have time, and on the y-axis, you have effective population size. Now, effective population size has quite a long-winded technical definition, but for our purposes here today, you can just take it to mean the number of breeding individuals in a population, even though the reality is probably a bit more complicated than that. And what you can see is that following the first appearance of koalas in the fossil record around 350,000 years ago, that's what the silhouette of the koala is there, there was a population decline and then a series of massive population increases following the emergence of our 5 major genetic clusters as distinct entities somewhere between 200 and 250,000 years ago, which all dovetails pretty well with the results of the ABC analysis that I presented on the previous slide. So that's always encouraging when you have different methods telling you the same result. But what's really interesting is that there was a massive decline in koala population numbers across all 5 major genetic clusters starting around 50,000 years ago. Now, the reasons for that decline are probably, well, almost certainly complex and multifactorial, but I doubt it's entirely unrelated to the fact that human beings are believed to have first arrived in Australia around this time, give or take 10,000 years. So what this would seem to suggest is that koala populations have proven quite vulnerable to both climatic and anthropogenic processes over time. All of which brings us back to our original question: does genomics, and specifically koala genomics, have utility as a bioindicator to inform ecosystem management? So the answer is a qualified yes, I would say. So I do think one of the primary advantages of genomics is that it can greatly enhance the utility of existing bioindicators, for example, by clarifying taxonomic relationships or differentiating between species that may be difficult to distinguish based on their physical characteristics alone, which is something Alan also touched on. But genomics can also provide ecologically relevant data that might not be immediately apparent from things like population surveys. For example, there's often not a one-to-one relationship between census population size, or the number of individuals in a population, and the genetic health of that population. For example, a lack of genetic diversity, which is the mechanism by which species and populations are able to respond to changing environmental conditions through processes like adaptation and evolution, has been linked to an increased risk of extinction. And lack of genetic diversity can also lead to what's known as inbreeding depression, which is basically a loss of biological fitness due to reproduction between closely related individuals, which in some cases, like that poor tiger down the bottom there, can be pretty obvious, but in other cases, it's a lot more subtle. So that coloured graph on the right there highlights the different levels of genetic diversity that we observed in the 5 major clusters of koalas. And basically what we found was that contemporary levels of genome-wide diversity, while they weren't significantly different between parapatric or neighbouring genetic clusters, on a continental scale, there was a clear geographical pattern of genetic structure in which overall genetic diversity declined with increasing latitude. So even though they have very high census population sizes, koalas in the states of Victoria and South Australia have relatively low genetic diversity, which may not only make them more susceptible to rapid environmental changes, it also makes them more prone to certain congenital conditions like testicular aplasia. And this is all information that you probably want to factor in if you're trying to do stuff like use the abundance of a single species or group of species as a proxy for the health of the overall environment. And finally, as we've seen in koalas, genomics can reveal past demographic trends in species with utility as bioindicators that can't necessarily be directly observed, but could also be very useful. Now, all that being said, one significant drawback to using genomics as a bioindicator is that it can take generations for significant demographic shifts to become detectable in an organism's genome, and those sorts of timeframes might not be compatible with the urgency of some management paradigms. And unfortunately, one major drawback to trying to use koalas specifically as bioindicators is that they're neither easy nor inexpensive to sample and survey. They're very shy, reclusive animals, so any sort of large-scale monitoring program would require a pretty substantial ongoing investment. But I think that there's definitely a lot of useful information about the relationship between climate and the environment that we can discern from koala genomics, as hopefully I've shown here today. So I'm just going to finish up by saying this is the reference to the paper that I was talking about if you want to look into it in more detail. And if you're interested in the more population genomics aspects of my talk, there's a link to another presentation I've given on that subject. And with that, I will say thank you very much for your time, and I will now hand you over to Professor Simon Robson. Cool. Take it away, Simon.
Okay, thank you very much for the opportunity to talk about bats as bioindicators. Looking forward to it. I guess when we think about bioindicators, we're thinking about a group, that can offer very rapid and efficient detection of something that's going on a broader scale, and that we can efficiently get an understanding of those broader processes by looking at a much smaller group. Of course, we have to be aware that all populations change over space and time, and then we have to think very carefully about how to select the best taxa. And we have to think about the ideal taxa versus a convenient taxa, because it might be one that we're used to working with. There are some guidelines out there. We need to be very careful to define what sort of environmental changes we're thinking about detecting, environmental biodiversity changes. Fortunately, there are some well-defined and rigorous criteria out there that we can use. I'll talk about in a minute. It's very important that we think about the time frame that we are interpreting and collecting information over. And it's probably pretty obvious that a suite of bioindicators is probably going to take the best approach. So here's sort of 6 of a variety of selection criteria that are out there. I'll mention them. I'll go back to these in a moment when we talk about bats, but it's things like cost, as our previous 2 speakers have discussed. Is it an affordable approach? Can you actually get in and measure accurately to get the information you'd like? Is there a historical data? So historical data can be very important for providing a context. Probably the trickiest of all of these, I think, is this sensitivity, responsiveness, or specificity. What the data are actually telling about broader ecological processes, as opposed to being perhaps a more sentinel taxa that says, look, something's happening and it's worthy to go and look at, but the change in this taxa alone isn't telling us what's going on in the bigger picture. So what is a bat? Now, what I've done here, I've just given a summary of some of the features of bats that I think are relevant. Very rich, about 20% of the mammals in the world. They have very diverse diets, so that's potentially useful if you're thinking about different models of nutrient flow through the environment. They range from solitary individuals to social individuals. Their roosting habits are quite broad. Perhaps one of the most interesting is that they can move a long, long way, and some cave species will have maternity roosts where individuals from females, the entire population will come back to a single spot and have their young, and that's going to be potentially useful for us. We know they serve important ecosystem services. They're sensitive to disturbance and pollution. They accumulate heavy metals, for example. They're very long-lived relative to other mammals of their size. And as we'll see, most of them echolocate, which is a particularly useful feature for us. So how well do bats match some of these? Well, public awareness is important. Unfortunately, poor old flying foxes get a flogging all the time, but there's a lot of effort going in to promote public awareness. And as the T-shirt says, either you like bats or you're wrong. You're in error. How about the cost of getting this information from? It's not too bad. There's lots of ongoing studies of flying foxes and microbat monitoring programs going on around Australia using visual counts, acoustic and video monitoring. So monitoring by listening to the echolocational calls or videotaping them. At the moment, as Beryl mentioned, I'm involved in a TERN project where we're monitoring the 7 vulnerable microbat species within Queensland. We've got detectors across the state, and that's less than $40,000 equipment set up, and that will run for many years. And an individual acoustic detector, there's a picture of one here on the right, you can get that and solar panels and all that for less than $1,300, and that can record bat activity every night. How good are we at getting the data when we capture bats? Well, it's fine for most species. So here's a couple of bats here where we can hold them in the hand. We have really good information. The taxonomy is solid. We can detect them and know what they are. But of course, there's always a couple of cryptic species out there that make it difficult to identify. And we need molecular approaches such as our previous speaker mentioned. Now, acoustic monitoring is become incredibly important. On the left is a sonogram of Hipposideros seminyi, microbat. Now the x-axis is the time and the y-axis is the frequency. And so this is a bat that's calling between 80 and 90 kilohertz. And you can see those yellow lines at the top is the call. This species, the calls are very invariant. They rarely change. It doesn't matter what's going on. We love these species. If the world was, if they were all like this, the world would be a different place. Here's Macrodoma guygas, and unfortunately, on the right of the screen, it produces very faint calls. They're difficult to detect. They overlap with other species, and also has these social calls, which are these things down the bottom. And I think it's fair to say that our knowledge of the variability within a species and across growth are making fantastic progress, but we still have some way to go. That for some of the species, we're still just recorded of bats flying around, but it has great potential. This is a new species of bat, was recently in North Queensland. It's a horseshoe bat flying. It echolocates at about 55 kilohertz. This recording was detected in a set of acoustic data. The individual went there, put out some traps, caught it. Brand new species, only known from a small land area of Australia, wouldn't have been detected without this technique. Video monitoring is a fantastic technique being used by a number of people throughout Australian groups. This is a project led by Terry Reardon and Paul Clissold and others at Narracrook Cape down in South Australia, where you put thermal video cameras up and you can detect bats flying out of their caves. You can videotape it. You can then start using some very clever software derived from missile tracking software that will count all the bats going out of that area and all the bats coming in real time. So it is an unbelievable resource. It gives you things like this is the last 12 months of data. We have on the y-axis is cumulative counts. This is in the 10,000s of bats. And you can see the bats use the caves a lot in the summer months and then decrease down in July. But it's really an incredible opportunity to be remotely providing real-time data on these bats. Historical data, well, as you can imagine, it varies for some species, but there's been a fantastic effort by Damien Milne and the Australasian Bat Society to collate all the information on Australian bats from museums and all sources and come up with their best guess of current and past distributions of bats. And anyone can access this on the web. And here it is, the records of the current possible and previous distributions of Macrodoma guygas, the ghost bat. So there's some great context there that we begin to understand. Now, the tricky one I've mentioned, I think, is making that link between what individual species or species or groups are doing and what's going on in the environment. This is a nice study from Europe. What we have here is we have measures of river quality. So the two graphs on the left column, the river quality is a measure based on abiotic features, and on the right, it's biotic features based on insect communities. They've divided bats into two groups, and one based on their ecology and their foraging. One group of bat increases with the river quality, one group of bat decreases. So again, we're getting these changes, but being able to link that to exactly what's going on in the environment, I think we still have a bit of work to do. It may be possible, but there's a bit of work to go on that area. But sometimes we can get some great results accidentally. So this was a lovely study, some individuals trying to understand the decline of ghost bats in Australia and found a very nice correlation, which is probably a causal relationship where Macrodoma was disappearing where cane toads were coming in. Now, surveying Macrodoma might not be the best way of assessing whether cane toads in the environment, but they are certainly telling us that information if we happen to have that data possibly collected by other means. Some of the bats are loved more than others. These are to Australia, Macrodoma rictus. They're a beautiful species. People like these bats, and so there's a reason why we have recommendations of bats as bioindicators. I think that some of the species of cave-dwelling bats, such as Miniopterus, are worthy of special consideration. They cover very broad geographic areas. The fact that they come back to maternity caves means you can monitor the population and the population growth, which is a very rare capability for any vertebrate running around or any animal in the world, I think. You can ID them remotely and relatively inexpensive, and they tick all those selection criteria that I showed earlier for picking taxa. And these are just the distributions of those three subspecies. And this is one of the Miniopterus. It might be upgraded from a subspecies to species. And this population, this is the sole distributed population, but we're pretty sure we know where all the maternity caves are, so we can potentially monitor the entire population. The other recommendation is that long-term basal studies are incredibly important. This is the results from Minneapolis caves, where we can see the population counts from the last 12 years. And here's a bit more video there. But the interesting thing is, and this, I just pulled this out of an email with Doug Mills, who's been running this project, indicating that even 13 years is just a starting point to begin to be able to put this into context. As I said, we see changes all the time. It's the interpretation of the broader context is the tricky thing. The other thing I think is we can make great progress by improving our ability to remotely detect back through acoustic means. And I'd just like to give an update here on the TERN Bats of Australia project. This is designed to provide acoustic resources to let people to improve their ability to identify bats by remotely monitoring bats in Australia and putting up an online library and presenting analytical tools. It's forming the basis for the sort of one-turn survey models for bats. Basically, open access, let people look at calls, identified and unidentified calls. It's almost, we're working on it, and it's not live yet, but so far we've received over 2 million bat calls, representing over 230 million bat pulses. So a pulse is an individual call in that thing. We're getting strong industry support. Titley, that produces software for analyzing bat calls, has produced a version that can be used online for people when they access the database. We currently have 70 echolocating species in Australia. And while it's not trivial to identify species based just on the call structure, certainly having this big data and the new tools we have, neural networks and all this stuff, machine learning, is going to be a great help. And so, and with that, I think I will just end and I'll hand back to Beryl. Thank you.
Fantastic. Thank you very much, Simon and Matthew and Alan. And we've been having a vigorous conversation throughout in the Q&A, and I think we're almost up to date. Simon, there's one for you about the bat maps, which I'll let you look at for a second while I ask a question. I'll just ask a question of Matt, if I can. Matt, do you think that the genetic differences in the populations that you've been talking about negate the use of taxa overall as bioindicators? Should we be cautious of the whole concept of bioindicators?
I mean, cautious, yes, but I wouldn't say it negates it. A lot of it depends on what level of distinction and differentiation you're interested in looking at, I would imagine. Certainly, at least in koalas, the level of genetic differentiation we find, while it does exist, it's not particularly large. So koalas are, you know, a single species. So in terms of that respect as a bioindicator, I wouldn't say it's an issue, if that answers your question.
Thanks. And there's a lovely question from one of our international viewers today, Sue, asking all of the panel, are people picking up evidence as yet regarding the genetic shifts in relation to changes in climate parameters, such as temperature tolerance, that is rapid selection in process? Does anyone want to start that conversation? You've got minutes, seconds.
Yeah, I mean, I think the short answer is yes. People are starting to detect those kind of rapid sort of genetic consequences of climate change and anthropogenic land management.
And Alan, do you think that we've made full use of our invertebrates yet? You say that they're powerful indicators for monitoring ecosystem change in land management.
Oh, well, no, absolutely not, Beryl. So I don't think they've been used as much as they could potentially at all. And so, you know, I talked a lot about their use in the mining industry. And so both here and overseas, that's been the case. But, you know, there's plenty of opportunity for making, you know, them having a valuable contribution to monitoring in all sorts of land management situations. And I suspect, back we talked about the potential of the genomics approach, and I suspect, once we've got that down working, it will be really accelerated the use of them, because at the moment, let's face it, most even professional ecologists have no experience with invertebrates and find them just too hard and too challenging, and so if we can obviate the need for detailed processing and identification of specimens, where very few people have the required expertise, taxonomic expertise, then I'm hopeful that will greatly accelerate their use.
Yeah, and Simon, while you're also answering that question that we have in the Q&A about the bat map, can you also comment, because you put prices on how much it's costing in Queensland for our pilot project? Do you think that it's too expensive to use microbats nationally as ecosystem bioindicators? So you've got two questions quickly.
No, I don't think it is. One of the great things we're going to get from this data of continuously monitoring is a much better idea of what the minimum amount you should be doing to get the answer we need to know. So that will inform us. But I think overall, it's a really, to be able to go and put out a machine for a little over $1,000 and come back a year later and get 12 hours a day is, I think, pretty incredible. The other question was about the bat map. So my understanding is that ALA and other resources can be a little sporadic, and they update names and this sort of stuff, and it can be potentially some errors. My understanding is that BatMap was created by individuals from the Australian Bat Society that have spent many years looking at bats in Australia. They got all the information from ALA, every information source that they could do, and then they came up with their best guess of what it is. So it draws on information from ALA, but I think it probably is the best spot for people to go if they want our latest understanding on where individual bat distribution is, based on all of those resources that were publicly available and privately available. So people donated all their data, and I think it's an incredible resource.
And the questions are pouring in now. Alan, a couple for you, if you could do them really quickly. Assume that you're not saying that genomics will put taxonomists out of business, and also are there indications that the eDNA is working to track community condition? With your microphone on.
Yeah, look, thanks for that question. Will it put taxonomists out of business? Now, the problem is taxonomists are out of business now, aren't they? And so, it's a real challenge, but I don't think so, because most taxonomists are not involved in these biomonitoring programs at all. So it is a very different situation, and maybe even it might provide more opportunities for taxonomists to validate some of the effectiveness of using genomics approaches. And and the thing is, if the genomics approaches are working here, and we don't need traditional taxonomy, we just have to accept that, don't we, for biomonitoring, if we want to have these invertebrate indicators really widely picked up and applied. So that's what I think about that one. So what was the other one? Let me see.
How well is ant eDNA working to track? And you've got
so it's at very early days, and so not just ant DNA, but just generally terrestrial invertebrates. So much of the work on environmental DNA has been in aquatic systems, and particularly fish and even invertebrates in aquatic systems, and very promising results. But in terms of particular soil DNA in land management, we've just really at the frontier of trying to work up that. I think one important point to make is the whole idea of a bioindicator is that you don't have to measure everything about an ecosystem to get some valuable information about how it's tracking. And the same goes within any particular indicator group. You don't have to get information on every single species to get valuable information on what they're saying about the environment. And so that's very relevant to these genomics approaches, because even if the genomics approaches are not able to identify every single species accurately, so long as they can distinguish different sites and provide information on how different sites are tracking, it doesn't really matter if they're not picking up every single species. And so this is an issue that, you know, we're looking at now. Is
Great way to end, Alan. Great, thank you. And I'm just going to share this last slide, and that is to say that we have got some more webinars coming up, and thank you very much, Alan and Matt and Simon. It's been really great, as evidenced by all the questions. And I'm going to leave you now, saying thank you, except I can't get my slide to move on. So thank you, everyone. See you again soon.
Thank you.